Today, I am honored to share the article titled Gut Microbiota-Derived Tryptophan Metabolites Improve Total Parenteral Nutrition-Associated Infections by Regulating Group 3 Innate Lymphoid Cells. Clinical nutritional support is recognized by Klinefner surgery as one of the 4 pivotal advancements in surgical practice during the 20th century. Surgeons regard total parenteral nutrition, TPN, as a key treatment method for rescuing patients with chronic intestinal failure, CIF. However, its use can lead to a series of complications, such as TPN-related infections, the incidence of which can reach as high as 42.05%. Exploring the pathogenesis of TPN-related infections is beneficial for improving the prognosis for CIF. The highlights of this study are that it reveals that TPN-induced gut microbiota imbalance reduces the response of intestinal group 3 innate lymphoid cells, ILC3s, thereby increasing susceptibility to infections, and Lactobacillus murinus and its metabolite indole 3-carboxylic acid, ICA, can promote ILC3s to secrete IL-22 by activating RORT, thereby playing a protective role. We found that TPN caused intestinal barrier damage and increased susceptibility to infections in 338 patients with CIF and in TPN mice models. Furthermore, fecal transplantation from TPN mice to normal mice indicated that the disruption of intestinal microbiota induced by TPN leads to intestinal barrier damage and increased susceptibility to infections. Through intestinal RNA sequencing analysis, we found that TPN leads to a decrease in intestinal innate immune function and a reduction in IL-22 expression. Flow cytometry analysis revealed that the decrease in both the number and proportion of ILC3s secreting IL-22 in the intestines is a significant reason for this. We further validated that the reduction in the proportion of NCR plus ILC3 cell subsets accounts for the decreased IL-22 expression levels. Furthermore, fecal transplantation experiments suggest that changes in intestinal ILC3s are attributable to alterations in gut microbiota resulting from TPN. We performed 16S sequencing and metagenomic sequencing on the feces of CIF patients and mice, respectively. The results indicated that the reduction of intestinal lactobacillus caused by TPN may be an important reason for the increased susceptibility to infections due to gut barrier damage. Among these, Lactobacillus murinus is a key strain. The single-strain intervention experiment found that Lactobacillus murinus can improve intestinal damage, reduce the levels of inflammatory factors, and increase the proportion of intestinal IL-22 plus ILC3s. Non-targeted metabolomics analysis suggests that ICA may be a key metabolite through which Lactobacillus murinus exerts its effects. ICA intervention can provide protective effects similar to those of Lactobacillus murinus. Aromatic amino acid transaminase, ARAT, is the key gene responsible for the production of ICA by Lactobacillus murinus. The protective effects of Lactobacillus murinus disappear after knocking out ARAT, demonstrating that ICA is a key metabolite through which Lactobacillus murinus exerts its effects. In vitro experiments, ICA dose-dependently increases the proportion of IL-22 plus ILC3s. Furthermore, it was discovered that ICA regulates ILC3s via RORT, and the use of the RORT inhibitor GSK805 can inhibit the effect of ICA. ChIP-qPCR, molecular docking, and dual luciferase assays further confirmed that ICA targets RORT to promote the expression of IL-22 in ILC3s. In summary, TPN-induced intestinal microbial dysbiosis reduces the responsiveness of ILC3s, and intestinal lactobacillus murinus and its metabolite ICA can enhance ILC3s function by activating RORT, protect the intestinal barrier, and reduce infection susceptibility. This study pioneers the revelation of the microbe metabolism immune regulatory mechanism, providing an important theoretical foundation for promoting intestinal rehabilitation therapy in clinical CIF patients and reducing susceptibility to infections.